Hello everyone. Today I am going to introduce you about 10 basic terms which are most commonly used in the subject microbiology. So let's start. The first I am going to introduce you here is culture. So as you can see here on the slide, I have included two pics. In first pic you can see the growth of microorganisms on the form of microbial colonies, and second picture you can see the growth of microorganisms in terms of turbidity in test tubes. The growth of microorganisms, whether it is on the solid, any kind of solid medium, solid base, solid support, and whether it is in the liquid medium as shown in the test tubes, is called as culture. So we most commonly use this kind of this term in microbiology laboratory, in microbiology subject, like this culture is of which bacteria? It is of E. coli. Store this culture in the refrigerator, take this culture, bring this culture. So it means what? The culture is what? Microbial group. Now we are going to second term. Second term which you should know about is pure culture and consortium. So what is pure culture? As you can see here in the pic A, I have shown that any kind of solution, if it is having same type of microbial cells like I have made here, cocci, circular in shape, round in shape, spherical, we can say, so then this type of culture will be of pure culture type, means it have single type of cell, microorganism cells. Now we are going to second type, B. B picture is going to show us that is any kind of solution, different type of microbial cells are present, different kind of you can see here cocci, spirali and rod shaped, and of course in next picture you can see here corresponding to it different color colonies. It means that it is having mixed type of culture. So mixed type of culture, for this microbiology we use term consortium. Now let's talk about the third term. It is medium or media. So microbiology media is kind of terms we use to represent what? Nutrient solution, but we use in the microbiology laboratory to support microbial group. Now fourth term, broth and agar. These are also other kind of terms which I use in microbiology subject. Broth generally refers to that kind of medium which is liquid in nature, like a picture you can see liquid medium is there, is nutrient solution is liquid. In the A picture medium is liquid, then we use a term broth. And if the medium that is going to support with the growth of microorganism we have taken in the laboratory is solid, then we are using a term agar. Why agar? Because in B picture you can see petri plates is here and solid medium is there, because same composition we can take any medium as we have taken in liquid medium, or in that we will be identify the solidifying agent which is known as agar. That is, we are saying that agar medium. So broth is a term for liquid medium and agar is term for a solid medium. Next term I am going to introduce you here is about sterilization. It is very important in microbiology. Everything we use in microbiology laboratory is for any purpose should be sterile in nature, and its need to process to become sterilized, and it is known as sterilization. And clear from the picture, if you have taken any kind of flask, you have prepared nutrient solution medium for microorganisms, and you are interested to isolate from food sample, pursue water sample, maybe that flask may be already where in some microorganisms, or that solution water nutrient and already having some associated with microorganisms, so they can contaminated, they can be called a contaminated. You are assuming that I have got this result from the milk sample, but maybe those kind of microbes I'll already present on the surface of the flask. So sterilization is very important, and sterilization we completely remove all living objects from what? from any kind of material. It can be glassware, it can be a medium. Picture is showing where organisms are present in, that is non-sterilized one, and where it is completely free of microbes, then we are saying now this sterilized. So sterilization we use in autoclave, hot air oven. Such kind of instruments are used in microbiology laboratory. So another important term which is used in microbiology is inoculation. Inoculation is actually the term we use in microbiology for the transfer of any kind of material in any kind of culture, any kind of sample into the medium. Like you can see here, I have shown a picture of solid sample. So I am going to transfer solid sample into a nutrient medium so that nutrient medium can support the growth of microorganisms present in the soil sample. Then this type of transfer is known as inoculation. Means I am going to inoculate soil sample into the medium. In the same way, when I am going to take store culture and I am going to revive it, means I am going to transfer the medium into the broth. It is called inoculation. Incubation. Next one is incubation. This is also an important term in microbiology laboratory. As a beginner, incubation term which is used to the time period which is required for the microorganisms to show a kind of growth which is visible to our eyes. Like if we have used any kind of sample, a water sample, and we have applied water sample on microbiology medium, and we can note results in the form of visible colonies. What is the number of microorganisms in water per ml of the sample? It needs some time. So that time which is required by a particular microorganisms to form a colonies to give us a visible growth, that time period is known as incubation period. For next important term is isolation. Isolation is very commonly used mainly when we are going to study microbial population of a particular sample. Like in laboratory we have received a soil sample, any pharmaceutical products, any food products, that we need to study for its microbial properties, how many bacteria are present in per gram or per ml of the sample. Then we will following the process. That process of study in microbial population form a sample is known as isolation. Next one important term, what every microbiology subject should be known as contamination. Contamination is a very serious concern in microbiology subject. Why? Because the contamination is actually the term which is used even some undesirable microorganisms are present in a tube, in a sample where we actually stored only one type of organisms. Like you can see in picture A, I have shown a next store culture which is present in a vial, present of store in vial number A. And in picture B, another tube is present having same culture. I am going to revive this culture and I am going to activate this culture. When I am going to take it out from the refrigeration, what I observed in A picture A tube, what happened? Circular shaped bacteria means cocci are present. On the other hand, in picture B, what I have used to tube number B, what you observe, the different type of bacteria are present along with cocci. Spherical rod were also present, means some undesirable microorganisms are or microbial cell have entered into the you need to be that whenever I am going to store my cocci culture was contaminated at that time. So I have done tube on any non sterile conditions what we were prevailing power there, they have contaminated to this culture, which is now not in our use. Contamination, again we can learn about the growth of the undesirable microorganisms in culture of our choice. So this is the last term what I am going to introduce you here is about subculture. Subculturing. Subculture or subculturing is a very important term which is generally used when we are going to make a second transfer of already grown in that plate. Why is a second transfer we are going to make? To purify the colonies or to purify the particular bacterial culture. Like you can see here in picture A, I have shown many colonies are growing here. And what we doing here, purify them to we will single we have to get a single colonies. Not only these colonies, we further multiply and same type of microbes or pure culture we can get here. In the pic B, suppose in first plate we are in next type of culture, when we are going to transfer a little amount of culture in such medium plates, what will happening, only that single colonies will be grow in on the other plate. So in this way the new culture is pure. It's mean of that purifying the culture which is used in the laboratory. And what can say second term transfer already grown in the uh, petri dish. So this was all about the basic terms what I have introduced you here, and I hope surely helpful and sureful to microbiology beginners. Stay connected through my videos, and thank you so much for watching.